Hi students, in today's video we will be discussing the topic bacterial conjugation. And bacterial conjugation is a unidirectional process. Here the transfer of genetic material occurs in a unidirectional way, and here a physical contact is maintained between the two mating bacteria. This process was actually worked out by three scientists, namely Lederberg, Hayes, and Woolman. And actually, before going to this process, we have to get a knowledge about some terms. That is, the bacteria which is having an F plasmid containing bacteria, they are called F positive. Okay, F factor positive, and they act as donors. And those bacteria which lack F factor or the F plasmid, they are called F negative, and they act as recipient. Okay, there is one more category, that is Hfr donor, that we will go through in the next slides. So if you are thorough with this F positive and F negative, we can move on to the conjugation. So conjugation, it can be of two types. It is based on the presence of two mating types. In the first category, an F factor possessing bacteria mates with a F negative form. That is, here conjugation takes place between F positive and F negative cells. So that is illustrated in this figure. See here, this is a donor cell, and this bacteria has a plasmid here. This is its chromosomal DNA, and this is the plasmid DNA. And this, see this bacteria, this is the recipient. It is not having any plasmid. So this bacteria which has a plasmid, we call it as F factor positive or F positive cell, and this is the F negative cell. F positive act as donor and F negative act as recipient. And now let's see what happens next. When this conjugation process occurs, actually this cell which is the donor or the F positive, it behaves like a male. So there will be a male like F positive donor and a female like F negative recipient. And there is a physical contact maintained here between this donor and recipient. And during the conjugation between this F positive and F negative, a hair-like appendages that we have already studied, that is the sex pili. This sex pili appear on the surface of F positive. You can see here, F positive cell has sex pili. So this sex pili comes in contact with the recipient. Actually, the main function is this sex pili, they help to hold the recipient cell in close contact with the donor cell. And we have gone through the details of sex pili, so I don't think I have to repeat all this here again. And the formation of the sex pili, it is determined by the genes which are present in this F plasmid. So when these two cells come in contact with the help of this pili, a conjugation tube or an interbridge is formed between these cells, and that is called, that intercellular bridge is called conjugation tube. So after the conjugation tube is formed, the F factor replicates. See, after the conjugation tube is formed, this F factor starts replicating, and the replication is in rolling circle model. And this one copy of this plasmid moves to the recipient cell through this conjugation tube. And as soon as the F recipient cell receives the F factor, the conjugation tube then separates out. See, as soon as this plasmid, one copy of this plasmid DNA comes into this donor cell, this conjugation tube breaks, and now the cell which was F negative now becomes F positive, and since it is F positive, it also forms its own 6 pili. So what is the result? In the initial stage we were having one F positive and F negative cell, and after the process we got 2 F positive cells. So that is the conjugation between F positive and F negative cell. And another category of cell is Hfr cell. In this case, the F factor, you can see the F factor here behaves like an episome. So episomes are nothing but the plasmid when gets integrated with the genetic DNA or the chromosomal DNA, then we call it as episomes. Here it is independent, it is not having any contact with chromosomal DNA. But if it comes in contact with this chromosomal DNA and integrates with it, then we call it as an episome. So in some cases these plasmids, they behave like episomes and they get integrated with the chromosomal DNA and thus forming a recombinant DNA. That is called Hfr, that is high frequency recombinant DNA. The strains or the bacteria which is having this Hfr arrangement, they will be showing high rate or a high frequency of recombination, and hence they are called high frequency recombination strains. And in the next, and this process is actually reversible also. This integration, they can disintegrate also again to form an independent plasmid. So this process is actually reversible. And now the second type of, if you are clear with all these, you can move to the another type of conjugation that is happening between Hfr donor and F negative recipient. In the first case it was between F positive and F negative. In the second case it is between Hfr and F negative. So here during the conjugation between this Hfr and F negative, the Hfr DNA of the donor undergoes rolling circle replication. And in this process, the circular Hfr DNA breaks open and becomes linear. See, this is an Hfr cell and an F negative cell, and then what happens? It starts replication, and the replication is rolling circle, and through the conjugation tube, the replicated portion moves to the F negative cell. And what happens next is after reaching there, the recombination process starts, and then we can have the result is a recombinant cell. So here a portion of the F factor occupies the leading end of the DNA strand and the other part occupies the lagging end. The leading as soon as this replicated DNA moves on to the recipient cell, after some time this conjugation tube breaks down, and so the plasmid DNA is not transferred fully, only a portion of the plasmid DNA is transferred and the other portion is in the donor cell itself. So only a part of the genome of the Hfr donor is transferred to the recipient cell, because before the process has completed, the conjugation tube was breakdown. So the DNA segment transferred to the recipient gets incorporated with the recipient chromosome, and in this way they are attaining the genetic variability. So this is the process. That is all.